सर आ रहे आए ना?
आदर सर, गुड आफ्टरनून. आदर सर.
Hello.
मयूर सर, एक मिनिट मी कन्फर्म
Hello,
करतो.
sir. Yeah, am I audible?
येस सर, येस सर.
Yes, sir. Yes, sir. You are audible, sir. Krishna, please continue.
Yes, sir. A very good afternoon to all delegate doctors. Thank you so much for joining for us today's webinar. So today's topic of webinar is dosage of reduction strategy in rabies management, role of the novel vaccine. Dear doctor, rabies is a world first novel three-dose anti-rabies vaccine, and with the great pride, we would like to inform you that rabies has successfully completed 1 year of launch with more than 40K animal bites victims are vaccinated, with 100% compliance recorded. So no case is ADR was registered. With this, it is my honor to introduce to you our today's speaker, Dr. Adarsh Sawat, specialty in Jeevan Rekha Hospital Director, related doctor. So now would I like to request Dr. Adarsh to initiate the talk, and later we will have a question and answers. So all the participants, please drop your questions on Dropbox. We will read out the all question after presentation. So Dr. Adarsh sir, please continue. Adarsh sir. हॅलो.
हॅलो, एक मिनिट सर, ते कॉल सरांचा कॉल चालू आहे. एक मिनिट.
ओके.
Hello.
Good afternoon, sir. Adarsh
Good
sir, good
afternoon.
afternoon.
Yeah, am I
Please
audible
continue.
as well as visible?
Yes, sir. You are audible.
Okay, great. Do let me know when are we supposed to start? I'm ready from my side.
Okay, you can start, sir.
Okay.
Hmm.
Also, do let me know, is the presentation visible or not?
Visible, sir.
Okay, let's start with today's presentation out here. Thank you very much to all the doctors out here for joining and for taking out your precious time out of your busy schedule and to attend this valuable CME out here. So as you can see on the screen, we'll be starting off with the Trabees, India's world's first 3-dose rabies vaccine out here. So basically, first thing first, what we'll understand is what is the need of modification in this vaccination. What is the need of Thrabies and what is the need of regime change out here. तो बेसिकली चल रहा है राबीपुर ले रहे हैं तो चलने दीजिए लोग दैट इज द बेसिक mindset by which people goes on with. So what is the need? The need is very, very clear. The first thing first is the fatality, and we cannot ignore the fatality of the rabies. We know, as we all know, there is 100% fatality rate when it comes to rabies and when it comes to when a patient lands up in a clinical setting certain. So we know there is 100% fatality rate. So precaution is the best thing to do here, and prophylaxis is the best thing that we have to carry out. So that said, second is the regime. The regime is very hectic, and that's why the non-compliance rate is so much higher in Rabipur. And third thing is the timing. Now, in today's world, the timing has got a great value. People and the patients, they value their time a lot. So by saying this, let's start off with this today's presentation out here. Myself, Adarsh, Dr. Adarsh Chawat out here. Thank you very much for taking out your time. And let's begin with today's presentation. This is Thrabies. And we can see, we will be covering these following topics out here. Let's begin with the you know the hero of the today's presentation. This is rhabdomyosarcomas. This is rabies virus out here, and as you can see, yeah. So as you can see, this is a zoonotic disease, and we have got 5 protein elements in the envelope out here. That is G protein, N protein, P protein, M protein, and L protein. And the protein that we are concerned with is the G protein. G protein is a major antigen in the formation of a neutralizing bodies against RABB. Okay, and here we can see the globally rabies deaths per year is 59,000 per year, which is alarming. And out of this, we can see 1/3 deaths are occurring in India itself. That is 20,000 deaths. And out of which 91% deaths occur in rural areas. So by this scenario, you can revise and yeah, as I said, rabies is 100% fatal, but 100% preventable with vaccine. So let's take a quick review of what the history goes. History of a vaccine out here. In 1885, there was 31 injection. It is impossible to make the compliance out here. And in 1911, we reduced it to 14 injections. And in 1976, it came came down to 5 injection with the regime of 0, 3, 7, 14, and 8 28 day. And since then, the surprising thing is since then, it has not changed at all. The regime, this is the their need of the society, that there has to be some change. we can't say that from 1976 it has not been changed. It has not been changed. Five injection was there in 1976, and the regime that we follow, we used to follow was 0, 3rd, and 17, 14, and 28, and still we are following that only. So yeah, the current challenges, as I am saying repeatedly, current challenges are the vaccines that we are using, used cell culture vaccines. And problem with this technology is it is 50 years old technology. Something needs to be changed. That is what I'm repeatedly saying. Something needs to change. And why the change is necessary? Because there is inconsistency in potency. There is there are stability issues, shortage of supplies because of so much dosage are required. 5 doses are required. There is shortage of supplies out here, and we can't ignore about the adverse effect of the vaccine. So adverse effects are also the one that needs to be updated out. Also, there is decrease of compliance, as I said. So let's go ahead out here. As you can see, the compliance rate after 3 injections, the compliance rate is getting reduced a lot. As we see, there is 41% decrease in 45% decrease in the rate of compliance, and there is not a 45% non-compliance issues with the fourth injection, and 54% non-compliance issues with 54 with the percentage of 54% non-compliance issues, right? So this is a major issues. Decreased compliance beyond third injection is the okay. Now just quickly let me know whether I'm audible and my screen is visible or not.
Sir, now you are audible as well as the screen also visible, sir.
Okay.
Mhm.
So are we good to go?
Yeah.
Okay, great. So as I said, let's come to this thing out here. Yeah. So what is the need? It is a first. So about the Trabees out here, this is the first protein subunit vaccine prepared by VLP platform technology out here, and so prepared by VLP platform technology out here. So as we all know, just 3 doses are required. This is the first 3-dose regime vaccine out here. And of course, due to 3-dose regime practice, there will be easy compliance, right? Due to this easy compliance and easy flow regime out here and less visits, the compliance will definitely increase. So, and one more thing, there is no reconstitution needed, and it is ready to use. So with no reconstitution and ready to use regime out here, this is the first 3-dose regime vaccine out here. So let's go to the VLP technology out here. VLP technology is it is virus-like particle technology out here. So as you can see, first the native virus containing the DNA material out here. So G protein is difficult to stabilize, and it is isolated as well as stabilized and purified by the Cadila Pharmaceuticals. then the virus-like proteins, virus-like particles are the protein complexes consisting of nanoparticles out there. So VLP are similar to the native virus particles, but do not contain any kind of genome, as we can see. So herein we can see all the antigens are available here. All the G protein antigen that is necessary, it is available. Just the viral molecule from the viral gene from the virus particle that we have removed, and then what we are left out with is virus-like particle, which is having a which is having an antigen particle with it, against which our body will be creating an antibody particles out. They cannot replicate, as we can see. There is no DNA material inside it, so they cannot replicate, as well as there will be no pathogenicity. So body will be developing strong humoral as well as cellular immune response out. So this is virus-like particle and VLP technology out here. So let's come to the clinical trials out here. So let's come to the clinical trials out here. So trial 2 results, the novel recombinant rabies G protein vaccine, the VLP based vaccine out here. So this is the study design out here. You can see we are taking the groups of 50, 50, 50, 54 groups out here, and we are taking one group of 25 people out here. So we'll be taking a 25 people. We'll be taking one group of 25 people, wherein we'll be checking the results of Rabipur vaccines out here. And we'll be taking a 4 group of 50, 50, 50. One group out of which 2 groups will be giving 10 nanogram dosage, and 2 groups will be giving 50 nanograms doses option. So out of that, 2 groups will be giving first and second dose only, that is on the 0th day and 3rd day. And out of 2 groups will be giving 3 doses, 0, 3rd and 7th dose. So here you can see the graph out here. On the 14th day, the seroprotection that we are developing with you can see the blue graph out here, blue bar, which is indicating the 10 nanogram dosage on 0 and 3rd day. And we are developing 100% seroprotection on the day of 14, on the day of 42nd, on the day of 19, on the day of 180th day. But on 180th day, the seroprotection is decreasing to somewhere 90%. And on 270th day, the seroprotection is decreasing to 85%. Then coming towards the second dose out there, second group, second observations, will be giving 10 nanogram dose to 0 on 0th day, 3rd day, and 7th day out there. And the results are same. Will be developing 100% seroprotection on the day 14th, day 42nd, and 90th day. But again, as you can see with the orange bar out here, the seroprotection is getting decreased up to 90% seroprotection. Okay, and on day 217th, the seroprotection is remaining still. It is not getting decreased to 85%. Again, coming to now the higher doses out here, 50 nanogram doses out here on 0 and 3rd day. As you can see with the gray graph out here, the seroprotection is up to 100% on 40th, 14th, 42, 90th day. as well as we are getting 100% seroprotection on 180th day. And on 270th day, the seroprotection is decreasing and it is becoming, it is going up to 85%. And now coming the full dose regime, you can see, you can easily make out with the yellow bar that it is getting displayed, 50%, 50 nanogram, we are giving the dose of 15 nanogram, and the full regime we are covering 0, 0th day, 3rd day, and 7th day out. With the yellow graph, with the yellow bar, we can see on 14th day, 42nd day, 19th day, and 180th day, we are having a seroprotection of 100%. And on 22nd day as well, we are having a seroprotection of 98%. And you can compare this with the dark blue graph, which is Rabipur regiment, which is 100% Rabipur regiment of 0, 3rd, 7, 14, and 28, 5 days regime out here. On the day 270, Rabipur is giving a seroprotection of 90%, 95, 90, more, somewhere around 90%, whereas the Travis is giving the seroprotection of 98%. So you can make out the superiority of the Travis in comparison to the Rabipur. So going ahead, yeah, trial 3 of clinical traits. Here our objective is to measure the safety and immunogenicity of the novel 3-dose recombinant nanoparticle G protein vaccine administered as stimulated post-exposure immunization. A randomized comparative control study was done. Randomized study was done. Multi-center approach was accompanied, and the phase 3 clinical study was done. So the study design is in front of you. Rabipur subjects, we have taken 267 subjects of Rabipur with 5 days regime was taken, and Rabies subjects we have taken 533 with 3 days regime we have taken out. So the total subjects we have taken is 800, and randomization that we have done is 2:1. So here, as I said, randomized comparative control multi-centric study approach we have engaged out here. We have opted for. The primary objective was the subject with seroprotection at 14th day post first dose exposure. The titer of RVNA we are achieving is more than or equal to 0.5% international units per ml. And secondary objective is subjects with seroprotection at 42nd day of first dose of test vaccine safety by monitoring the subjects with the adverse effect. So here you can see the efficacy. Efficacy is more on the 14th day. By the graph, you can this thing make out by the graph. You can see the efficacy is around 99%, 99.24%. By dark blue graph, you can make out the efficacy is around 99.24% of Thrabies, and the reference vaccine is 97.72% on the day 14th. And on day 20, on the day 42nd, the efficacy is around 98.69%, and whereas the reference vaccine is having 100% efficacy rating. Now coming to the main objective of the study of the clinical 3 trials out here, the safety and the adverse effect. It was considered the adverse effect. This is the major thing to note. Here the adverse effect is only 99%, as compared to the 17.2% adverse effect of the Rabipur. So this is the notable thing to note is Thrabis having adverse effect of only 99% as compared to the Rabipur, which is having the adverse effect of 17.2%. And with the p value of 0.0032, so the null's hypothesis, as we all know, null's hypothesis which is required is 0.05. To consider the null's hypothesis, we require 0.05%. But here you can see, as you can see, the hypothesis, the value of p is less than 0.05%, less than 0.05. Therefore, we can reject the null's hypothesis over here, and we can establish the significance of the study subjects out here. So we can establish the significance of the study subjects that we have taken, the comparative study subjects that we have taken, and we can reject the null's hypothesis, because here only 3%, as we can see, p value is 0.0032%. So coming to the local adverse effects is localized pain with the value of with the Thrabis showing 7.69%, as compared to rabies, it is lesser. Again, coming to the p value, it is 0.0075. So here also we can reject the null's hypothesis. Redness, coming to the redness and swelling, again it is lesser than Rabipur, but here you have to consider the null's hypothesis, because it is 0.6635, and in case of swelling it is 0.1559. Now coming to the systemic adverse effect out here, the fever, it is markedly less, which is only up to 0.38% as compared to Rabipur, and with p value of 0.1931, we have to consider the null's hypothesis over here. Also, ear pain, we cannot ignore the near ear pain out here, and the vaccine is having some kind of association with ototoxicity. So I would request to have with the patient with some kind of ototoxicity or some kind of ear related some kind of ear related this side effects, there has to be some kind of screening needs to be done after before giving therapies out here. But with p value of 0.0860, we cannot reject null's hypothesis over here. And headache is somewhere around 0.03%, p value of 0.86. So significant amount of null's hypothesis. We cannot reject the null's hypothesis over here. Same goes with urticaria, joint pain, and nausea. We didn't find any kind of joint pain, any kind of nausea related to Thrabies. Some kind of urticaria up to 0.19% was recorded, but the p value of 0.8607 was noted. So null's hypothesis, we cannot reject the null's hypothesis. So in conclusion, let me conclude today's this thing. In conclusion, I can make out that the results were consistent across the different age groups and genders. Observed differences 1.72%, with the pre-specified margins of 4% out. And I can make out non-inferiority for seroprotection, primary end and point, primary endpoint, and day 14th as well as 42nd were met. test and comparator are identical, and seroprotection rate both on days 14th and 42nd, as you have observed earlier. So, in conclusion, let me finally conclude the study that we have taken out here. Rabies was not inferior to the rabies vaccine, to the reference vaccine, but in contrary, it was somewhat superior only. But there was also in clinical trials of a third phase, we can make out less adverse effects compared to the reference vaccine. Also, so in conclusion, let me conclude out here. Novel 3-dose rabies was found to be safe, immunogenic, and was comparable to 5-dose of WHO pre-qualified vaccine out here. And you can see the achievements of rabies out here by Economic Times, Pharma Innovation of the Year, by Innovative Product of the Year. We have been awarded Innovative Product of the Year, the Golden Globe Tigers Award. I would like to thank you. I would like to present a vote of thanks, sincere vote of thanks, and thank you very much for calling me on behalf of Cadila, and thank you very much for giving me the opportunity for presenting the presentation. I thank all the doctors present over here who were able to manage their valuable time and take out their time, and they could come and attend today's CME out here. Thank you very much. I am taking off. This is Dr. Adarsh Chawat. Thank you very much. Take care.
So, on behalf of Cadila Pharma, I am Krishna Kasar. Thank you so much, Dr. Adarsh, for your informative talk. We are truly appreciated your efforts. It's a very excellent session of today's webinar, sir. So last but not least, we would like to inform you that Thabis has won 2 prestigious awards in innovative category, that is Economic Times of India Pharma Awards and Global Golden Globe Tiger Awards. So thank you all delegates who are joined here, and once again thanks for Dr. Adarsh for very excellent and interactive speech, sir. So there are अगर किसी का क्वेश्चंस रहेंगे तो सर पूछ सकते हैं सर. सर मेरे हिसाब से अगर किसी का क्वेश्चंस नहीं रहेंगे क्योंकि चैट बॉक्स में नहीं है, तो हम अगर सेशन को यहाँ पे एंड कर सकते हैं सर. मयूर सर, हेलो.
हाँ कृष्णा, कोई
Session end कर सकते हैं.
क्वेश्चंस नहीं रहेंगे तो सेशन एंड कर सकते हैं.
Yes sir, yes sir. Tech support, you can end the
जी
session.
सर.
End कर सकते हैं सर.
ओके सर.
Okay sir, thank you very much for your support.